and this is our Google Hangout. Today is one in a series of Google Hangouts with Sigma Xi's distinguished lecturers. This particular Google Hangout is sponsored by the Research Triangle Park chapter of Sigma Xi. Anyone watching the Hangout, you can submit questions at any point. They will show up on my screen and I'll then ask Dr. Hildebrand about them. And we will also be live tweeting this Google Hangout with the hashtag AmSciGHO. Today we have with us Dr. John Hildebrand of the University of Arizona in Tucson, who's going to tell us about his research on insects, in particular their brains and their sense of smell. Dr. Hildebrand, thank you for joining us today. I was intrigued to see that the title of one of the talks you have been giving as a distinguished lecturer is How an Animal Thinks About Odors. Can you sort of set the scene for us for that talk by explaining how you came up with that formulation?
Yeah, I came up with that title as a way of attracting people to come to the lecture. What it's really about is how olfactory information is processed in the brain and how it underlies the natural behavior of the creatures. But it's activity in the nervous system in the brain, so I took the liberty to say how they think about odors so that people would find the topic appealing.
I understand. So, and you did take the caution of putting think in quotation marks to make clear that insects do not, for example, have little thought bubbles in their head saying, I smell a lily at 2 o'clock. But you wanted to make
but to be clear, I don't think we know that. I mean, we assume they don't. I always respect the little creatures and say what they experience. We assume they don't think and have little thought bubbles. I think it's better to be cautious and not make any assumptions.
Fair enough. Okay, why don't you tell us a little about one of the insects you have spent a lot of time studying, the hawk moth, Manduca sexta. What does it look like? Would we recognize one if we saw one? Where do they live? Would a human in the course of a normal urban or suburban day have anything to do with one?
Yeah, well, probably many people, they may not realize that they've seen them. Let me start with their caterpillars, the larval form. Anybody in the United States who's tried to grow tomato plants in their garden and has had a great big caterpillar eat the plants, in all likelihood has had that Manduca sexta caterpillar working against them. That caterpillar is a green a creature the size of a pretty good sized cigar, and it's voracious. A single one can eat all the leaves off of a single tomato plant. So that's one way people have seen them. As an adult, they're a big moth. They have a wingspan of about 4 inches, and they behave a lot like hummingbirds. So people seeing them at dusk or even later in the evening during the warm season may think they're seeing a hummingbird, but in fact they may well be seeing Manduca. It hovers over flowers to feed on nectar. The females hover over plants to lay eggs. And because it's usually at night that they're active, we don't see them. They're hummingbirds. There's a relative of Manduca sexta that's a similar moth, and it's actually called the hummingbird hawk moth, because just acknowledging the fact that they behave so similarly to hummingbirds. But you asked how they're distributed. Manduca sexta is found in nature from northern Argentina all the way to Canada. Excuse me, I'm having a cough. And they're found in most parts of the United States. They're active in the summertime, starting late spring and through into fall, and they are not seen during the cold winter months. Tomato plants are one of their favorite plants to eat, but that's not the only one. They eat tobacco plants and other plants related to tomato and tobacco.
Where do they go in the winter?
Well, the adults are dead by wintertime.
Okay.
They only live a short lot, long enough to eat and reproduce. But what overwinters is their pupal form. So, this is a complete metamorphosis. The eggs hatch, little caterpillars emerge. They go through 5 molts, getting bigger and bigger and bigger, till they wind up that big cigar-sized thing. That then finished with larval development, turns into a pupa. Sorry, I have to cough again. This is the problem with dry air.
Mm.
And it's the pupal form that actually overwinters underground. They emerge from the larva underground, stay there until the ground warms up, and then they turn into a moth that starts flying around and doing what moths do. Sorry, I hope this will stop.
When they, how do they get deposited underground? Is the adult moth digging underground?
No. So the pupa comes from the larva. The larva grows. I'm sorry, we don't have a diagram here, but the larva grows from a tiny thing to a great big cigar-sized thing through a period of about 20 days of eating voraciously and molting. Each time it molts, it takes a big step up in size and winds up a big thing. At the end of that period of growth and molting, the larva turns into a pupa, but it does that after crawling off the plant, digging into the ground, and therefore in the warm months, that pupa then metamorphoses back. When it gets cold and the days get shorter, the pupae that form then go into a kind of hibernation. It's called diapause, and they spend the whole winter underground, only to become activated and turn into a moth that climbs out later in the spring when the ground warms up and the days get longer.
Okay, so it sounds like partly the story of the very hungry caterpillar, and then later not so much. But the idea of eating more and more and getting bigger and bigger, I wonder if that was the inspiration for the children's story. I won't read that to any grandchildren again in the same way. I'll say, it's possible. It didn't turn into a butterfly, but I digress.
When you do read it to kids, make sure you tell them about Manduca, because the word Manduca means glutton.
Oh, that's wonderful.
So it's clearly
So there is a connection there. That's great to know.
Yeah.
Okay.
Yes.
Now, you have written that Manduca sexta is an excellent model for the study of the neurobiology of olfaction. And I wanted you to tell us why that is. What makes a good animal model for the study of something? Why is Manduca sexta such a good model for the neurobiology of olfaction?
Okay, there are several points to make about that. The first one is that insects like Manduca depend on olfaction for practically everything they do. So if one wants to study the neurobiological basis of olfactory-dependent behavior, it's good to use an animal that does everything on the basis of olfaction. So finding mates, finding food, finding places to lay eggs, all of that depends on olfaction. So that's point number one. We know a lot about the behaviors that depend on olfaction, and it's most of their natural behaviors. The second point is that much to the surprise of people who don't know about this, the way the olfactory system is organized at the cellular level, from receptor cells that detect chemicals in the environment all the way through the brain, through information processing circuitry, the layout and organization of the olfactory system of an insect like Manduca is very similar to that of a mammal like us. So that's what we mean by a model system. It's a system that bears many resemblances to the systems in creatures like us, but there's no ethical constraints on studying them experimentally as there would be with humans. Moreover, although the layout and function of the olfactory system is similar to that of a mammal, their olfactory system is much simpler in the sense of far fewer nerve cells. So instead of millions of cells in the pathway, there are thousands, even hundreds in some parts of it. So it's more comprehensible. We can actually analyze the circuitry and analyze what happens to information detected by the receptors in the periphery as we trace the information through the brain in a way that's much more facile and I would say powerful than we can in a more elaborate vertebrate animal. So those are some parts of the answer. And Manduca, well, I chose to work on Manduca about 45 years ago. And one of the main reasons is that it's a big insect. And its nervous system, its brain, is big too, much bigger than that of many other insects. And it's good because the nerve cells in the system are also bigger than those of other insects. And although we could study other insects, and many people do, to learn about olfaction, the way we proceed in our work, having big nerve cells in relatively smaller numbers is a big advantage, and Manduca offers that to us very robustly.
Okay, well, is there a problem looking at Manduca as a model for the study of olfaction, for the neurobiology of olfaction, comparing it to animals that rely much less on their sense of olfaction for the information they need about the world?
No, I don't think so. What I tried to say when I introduced the topic is that the advantage of using an animal that depends heavily on olfaction is that we know a lot about the behaviors that are served by the olfactory system. And I'm a neurobiologist whose point of view is what we call neuroethological. I'm interested in nervous systems because of what they do for the animal. I'm not interested in them because of disease and so forth. So to understand what a sensory system like an olfactory system does, I want to understand how it serves the natural behavior of the animal in its real world, going about the business of living. So finding an animal like Manduca that really heavily depends on olfaction offers a rich array of opportunities to study the olfactory basis of behavior. And it also informs us about what that olfactory system is meant to be detecting and what it's meant to be responding to. This is a very fundamental point, so I'm glad you pushed the button. You know, when you study a sensory system like the visual system or the olfactory system or the auditory system, a sensible way to study it is to ask, what does the animal use it for? In other words, what stimuli are meaningful to the animal, because that's what the system evolved to deal with. It's not as sensible to do what many people in the past have done, which is to say, what do you think of this stimulus that I'm interested in? Because the animal may not be interested in it. So this is a very powerful message. It was the discoveries of how our visual system works, but it's also true for other sensory systems.
Well, that makes me wonder if in humans, for example, olfaction is offering us more information than we may be using because we have other senses that we rely on more, or perhaps if we're taking in more information through olfaction than we're conscious of, and it just gets mixed in there with other information, and we don't think of it as tracing back to our sense of smell. Do you think that might be possible?
Oh yeah, I like that question too. I think there are a couple points I'd like to. You know, we grow up being told that, well, our olfactory system isn't very good. Dogs have a much better olfactory system, and that's true. But the truth is, and it's very apparent now, and as more than ever before, is that our olfactory system is much more powerful than we thought it was. That's point number one. Point number two is really to your question. We assume that we know what we're using our olfactory system for. We assume that we're conscious of everything it does. But what fascinates me is the very great, in addition to the olfactory stimuli that we're aware of, that we are conscious of, that olfaction could be playing many roles in our lives that we're not aware of. There's much evidence for that, which we could go into if you wish. I mean, one example is evidence that olfactory stimuli that we're not conscious of actually are important in bonding with potential mates. So in other words, maybe play a role in choosing a date.
I heard about an experiment with men's T-shirts, I think it was.
That's
Is that
the
one?
study I'm thinking of.
Yes. That
Yes.
I think it was in the case of couples who had already chosen each other. So the question was whether they could recognize the used T-shirt of their mate as distinct from that of other men. Am I getting that right?
Yeah, well, that's one version that don't involve people who know each other, but showing interesting correlations. This is kind of technical, but correlations between what's called histocompatibility genes or loci in the genome and attract of the scent from a used T-shirt. This gets kind of complicated, but it's a fascinating story because we think that we choose partners based on how they look and how they make us laugh and so forth, but our sense of smell may even know about. We weren't conscious of it.
Right. Well, what do you make of the fact that in humans we have this one organ that's kind of not formulated all that well for taking in smells from a wide range of directions? Do you think that might be part of the reason we play down our sense of smell, or are we going too far afield from your research?
I'm not sure I understood the question. You're talking about spatial.
Let me go back to let me go back to Manduca, because that was really where this question is.
Okay.
If I understand right, the antennae are the primary olfactory organs of Manduca sexta.
Yes, we're told when we're kids that antennae on insects are feelers, and in many instances that's true. They play an important role in touch, but in a moth like Manduca, the primary function of the antenna, the part of the antenna we can see, is olfaction. It's the primary olfactory organ. It's not the only olfactory organ. I mean, they also have other olfactory organs that are rather specialized, and that's true of other insects like flies as well. But the antenna is the primary olfactory organ in moths.
Okay, and they can move those antennae around. Is that better to home in on particular scents that they need to know about?
Yeah, so moths don't actually do that.
Okay.
You're talking about moving the antennae kind of like that.
Yes.
Cockroaches and crickets and many insects do that. Moths don't do that. They can move their antennae, but they basically hold them in a resting position or in an alert position when they're flying, and they hold them like this as they fly, without moving them very much. So it's a different strategy of using them. It's like having just two elongated noses on the two sides of the head. But the basis of your question, the question is, do they get directional information because they have two of those appendages? I think that's part of what you were thinking. And the answer is yes. They can tell whether a scent is coming from the left or the right. The question, though, goes on, how much do they use that information? What is it important for? And that's much less clear. The information is kept separate as it goes into the brain, so that means they do get different information from the two sides, but it's not clear that it plays a big role in behavior.
Is there work that's going on to try to trace how they use the possibly directional information from one antenna versus the other? In other words, would it be possible to knock out the receptor cells in one antenna and see how they do with just the other one?
Yes, wonderful question again. Experiment in the past, you can knock out the input from one antenna completely, and the moths still fly and behave more or less as they should in response to, for example, but having only one functioning antenna causes some problems in the control of flight, because odor isn't the only thing the antennae detect. The antennae also have their, that is at the special mechanosensory
insect in the insect brain compares with the olfactory bulb in the mammal brain, and I think that's going to be a long and descriptive answer.
Well, I'm going to keep it really brief. Suffice to say that the antennal lobe, which is the lobe in the brain of an insect like Manduca, is very similar to the olfactory bulb in a mammal or other vertebrates. It's just a lot smaller and has far fewer elements, far fewer cells. But other connections among nerves, the synapse, the way they're organized, the way information comes in from receptor cells in the antenna and is processed through the circuitry in the antennal lobe, all of that is similar to the way information from the nose or olfactory bulb. It's just simpler because there's so far fewer cells. So we can sort of understand what's going on more readily than you can more elaborate olfactory bulb. It's a matter of scale. People often talk about invertebrate nervous systems as being simple. They're in a sense of easy, but they're simple in the sense of numbers. Far fewer cells, far fewer synaptic connections, and therefore much more readily analyzed with the tools of neurobiology. So that's really the attraction of the system as a model. But as I've stressed earlier before the break, it's a model because the way it's set up, the way it is so remarkably similar to the way ours works. So what we learn in the insect is directly applicable to questions about how in vertebrates. I find it amazing because, evolutionary biology tells us that had a common end with insects about 600 million years ago, and yet here we have very similar olfactory system.
I guess it may be a case of if it works, stick with it. If it's a system that's working well,
yeah, I like that.
Keep using it.
Okay. The way people think about those things is either that if it works, stick with it. In other words, conservation in evolution, or convergence. That is, independently coming to the same strategy because it works so well that it's been discovered. Mulch is true, but I kind of like the one you mentioned. I like that it was working really well long ago, and so it's still working.
I guess it would be difficult to prove either way, but can you think offhand of an example of something that was studied and came to be understood in insect olfaction that then prove to apply or to address a question in mammalian sense of smell?
Yeah. I wish I had diagrams here. So the olfactory bulb in a vertebrate and the olfactory lobe or antennal lobe insect in structures that are called glomeruli. These are ball-shaped structures that are very densely packed with the processes of nerve cells and synaptic connections among them. And one of the questions that have interested people like me to get us into this function of those olfactory glomeruli, these little ball-shaped condensed structures within the antennal lobe or the olfactory bulb. We knew since the 19th century that the primary olfactory center in the brain of just about any animal, insect, invertebrate, has these, but there were speculations about what their function was. We didn't know for sure. So one of the things that was first shown in insects and later shown in vertebrates is the fact that those glomerular mailboxes receiving information about a particular segment of the olfactory world. What do I mean by that? Well, you have to realize that olfactory stimuli, odors, they are made up of huge molecular compounds. We can detect millions in the olfactory system. Insects deal with large numbers of olfactory stimuli too, but the numbers are much smaller than what we deal with. And in fact, in one particular case, that is what's called sex pheromones. These are the molecules that attract a case of moths. In that case, there's a specialized glomerulus for each of the compounds in the pheromone. So it's one-to-one, and a particular glomerulus. And discovering that was the first little glimpse through the keyhole that taught us that glomeruli are mailboxes that are doing olfactory stimuli. The olfactory stimuli aren't represented everywhere. They're represented in a pattern of projections of receptors. And that turns out to be true in mice and humans and snakes and fish and every insect that's been studied. It's a very basic principle, first demonstrated solidly in insects.
Great. Okay, something I saw in the notes of your talks that I was dying to ask you about is the Manduca BioBot. Did you and colleagues constructed or are in the process of or are thinking about a Manduca BioBot?
Yeah.
What is that for?
Okay. That project is no longer happening, but it started back in the last century by a program director at DARPA's Advanced Research Projects Agency. Very interesting research agency that does a lot of blue sky research, thinking of the future. And they were very interested, as one of their first adventures in biology, in what they call biological systems. Of course, the nervous system is a biological control system. And I was approached to get involved with that program to see if we could use the of an insect to a robot, let's say a little wheeled vehicle. That way you interface the living control system with the mechanical engineered device, basically putting the moth in the driver's seat. So if the moth is interested in a particular odor, the attraction to the odor would end up steering the vehicle toward. That's a biobot. It's a hybrid between a robot. There are still many people working in that field, especially, for example, in Japan. But we were, I guess, kind of pioneers, and we had some significant success with that. We had subsequent DARPA contract to do something a little differently, and that was to interface electronics with the nervous system and get a flying moth to go in directions that we wanted it to go. In both cases, what DARPA was interested in was using the insect to help with what it's gathering, to what chemicals are in the environment in various places of interest, for example, to troops in the field. So enlisting moths as aids to intelligence gathering. I thought it was a pretty advanced notion. And there's still a lot of research like that going on.
That sounds like it could have all kinds of applications, maybe for tracing epidemics, detecting dangerous chemicals that are at too low a concentration for people to perceive, but that could still hurt them.
Right.
Okay.
Absolutely. Well, that's exactly. Drug interdiction, for example, is another area that we were interested in using the same.
Huh. So these would have replaced maybe the dogs that sniff the baggage of passengers returning from various countries. They would just be flying around and people would think, well, what a nuisance, but actually those moths would be doing.
Yeah, that's right. Yeah, that's right. I mean, it wouldn't replace dogs. Dogs are awfully good, but a moth flying around wouldn't be noticed.
Right.
Not to the degree that I would say right away that this is not yet deployed as a tech in battle zones. But
yes.
But this what DARPA does is to explore possibilities, kind of push the envelope.
Well, if we do see them sometime in an airport, we'll know it's doing a job. Don't pet it. Don't try to feed it. Okay, let me ask you about another study in which, if I understand this right, you transplanted female antennae onto male moths. What were you trying to find out there, and what did you find out?
Okay, that's a project we did quite a long time ago. We actually did it both ways. We transplanted male antennae and female antennae onto males. This is in the adult moths. And what we were trying to find out in that case was what influences the development of the olfactory bulb, the olfactory lobes in the brain, the antennal lobes. They are not the same in males, sexually dimorphic, and we were interested in what controls that sexually dimorphic development. So in order to, one approach was to transplant the primordia, the precursor, back in the larva, back in the caterpillar stage, and then allow those to go through metamorphosis and develop into moths. And when we did that, the graduate student who did that with me was very successful. The wrong antennae on them. There's a fancy word for that. This is an antennal gynandromorph. It means it's a male moth that has female antennae or a female moth that has male antennae. Well, not only did the brain change commensurate with which antenna was present, so the antenna of a female that had a male antenna on it now looked like a male antennal lobe, and vice versa. But also the behavior was changed. The behavior of the moth, and one very specific example of that I referred to here, the detection in response to sex pheromone. That's something that in Manduca, only the female releases the pheromone. The male moth detects the pheromone and then flies in a way that brings him to the source of the pheromone signal. Females don't do that in Manduca, but when we made the gynandromorphs, so we now had a female animal that just had male, that now behaved like a male and flew to pheromone, something that a female would never do. So this was quite exciting to us. The bottom line of what we learned from that, well, there are several things we learned, but one of the main things is that the sensory nerve cells in the antenna, when they grow their axons, their processes into the brain, those growth processes, when they reach the antennal lobe, exert a very powerful control over the development of their target. So whichever input is growing in kind of governs what the antenna lobe was going to be, they're male or female in its organization and function.
Fair
So that
enough.
was that question, but it's a very basic question, but really interesting from a developmental neurobiology point of view.
Yes, it sounds like the antennal lobe calls the shots there in terms of what the developed animal will eventually do.
Right.
Now, here's a possibly troublemaking question. What would happen if someone had tried to transplant one female antenna and one male antenna early in a developmental stage? You'd have to transplant both of them because otherwise you would have a transplanted and a kind of native grown. But what if the moth was getting contradictory signals? Did anybody try this, or would they?
Well, that's an interesting question. Actually, we've never done the experiment the way that question is formulated. But what we did a lot was to transplant one antenna. So we would end up with a female moth that has its own antenna on one side and a transplanted male antenna on the and so that's a little bit different experiment, but it's still contradictory information. One antenna lobe is getting female specific information, and in that case, when we tested them in the behavior that I just described, the orientation to sex, it's the male version. It's the male side of the animal's behavior. The animal now behaves like a male, and it flew to pheromone. We can do the opposite thing. We could put a female. In that case, the behavioral change is less obvious. But the behavioral change, and that is that the male moth now is more something that matters to females for egg laying than a normal male. So this is an experiment that kind of works in both directions, very obviously in one direction and pretty well in the other direction as well.
Okay, we have a question from someone from the audience. That is, I believe it's true that the olfactory sensory flow does not pass through the thalamus, as is the case with other senses, and therefore is not subject to declarative recall. This would be in mammals, in humans specifically. Thus, one cannot recall smells in the same way one can recall visual senses. How do you view that in an evolutionary sense?
Well, let me just say, you were breaking up a little bit, so I'm not sure I got the question completely right. But what I did understand, I think, isn't quite true, that of the 5 primary senses, is the one that doesn't first go through a thalamic relay. It does have secondary projections to a thalamic relay, but in fact, what happens is that the olfactory lobe has privileged access to the limbic system and the hippocampus. Those cortical structures input from the antennal outputs, the olfactory inputs. And so, in fact, olfaction is a very powerful and evocative memory. Most people who have a functioning olfactory system have had the experience of walking into a room where there's a scent of a pipe tobacco or a cologne or bread baking. Very profound memories triggered by that scent. That's because of the privileged access of olfaction to the parts of the brain that deal with memory, or that are specialized to deal with memory. So also emotion, the limbic system. So in fact, olfaction really is very important for memory, event-related memory, and emotion-laden memory. It's hard to make a comparison with how insects are organized because they don't have a limbic system, and they have their own structures in their brain that may play parallel functions, directly relatable.
But is it possible to deliberately recall a scent in the same way that we can conjure up in our minds what something or someone looks like. I think that's what the question was referring to. Declarative recall. You passively, yes,
I didn't
they're
get that.
very
Yeah.
strong, but can you kind of actively recall a smell? Is that possible?
Oh, okay. If that was in the question, that's when you were breaking up and I
Ah,
didn't hear it.
okay.
That's a good question. I think it's harder to conjure up, a concept of a smell than it is, for example, to imagine a face. Uh, that's true in my experience. Now, I don't work on human memory, so I'm not an expert on that. But I think one of the problems with smell and flavor, which is mainly smell, is that we have such a poor language to describe those sensations. With a face, we can talk very specifically about the features of a face and how they look and the colors, but we don't have a good language for. You have to make it all by analogy, and it's sort of like citrus. So conjuring up a scent is difficult, I think, partly because we just don't have a good way to categorize it.
Interesting. Okay, just to turn briefly to the other side of your research on insects, you do also study insects as vectors of disease, and in fact, they're a very significant vector of disease. Um, Among the information you sent me was a chart showing mosquitoes, if I have this right, as the most dangerous animals on Earth. Is that correct?
Yeah, and I use the title "The Most
Yes.
Dangerous Animals" to attract people to my audience, and I ask them if they think it's the great tiger, or what do they think is the most dangerous. People usually guess it's humans, but in fact, mosquitoes are responsible for more human morbidity and death than any other creature on the planet. And that's if you add up all the viral, bacterial, and parasitic diseases that are spread by mosquito vectors in the course of humans. The total toll that takes on human life is enormous. The most obvious primary example being malaria, but it's not the only one. There are many other diseases. Everybody hears about West Nile, the viral illness called West Nile disease, about dengue, which is in parts of the United States, chikungunya, the new disease that's just started to show up in the Caribbean, and I could go on and on. There are just so many diseases that are vectored by mosquitoes, and we don't get an awful lot of. So, just in their numerical toll on human health and welfare, they are the most.
Oh, that leads me to another question. Why do you think it is that the actual magnitude of the danger to human health that mosquitoes represent is far greater than our awareness of that danger? Or maybe, am I just asking this from the perspective of someone who lives in a fairly urban Western environment where we have mosquitoes more or less under control? Is there more awareness in other parts of the world of how dangerous mosquitoes can be?
Yeah, so you put your finger right on it. If we lived in tropical Africa, or would have a very different view of them than we do in this country. I mean, here most people view them as a nuisance, which they are. But in those parts of the world, the toll on humans is enormous. Uh, in Africa, the only thing that really works sleep under pesticide-treated, so that the mosquitoes have a hard time getting to the people for blood feeding. I mean, you have to wear the mosquitoes in order to know that you're going to have to sleep under a bed net. Prominent in their thinking. In this country, we don't have malaria. We have troublesome mosquitoes, and I'm going to be more and more aware of it. It's not a pleasant disease. The border in Mexico, and it may there are people right here in my university trying to figure out the problem here in Arizona yet. But I also mentioned chikungunya, another viral disease spread by mosquitoes. That has come from the eastern part of the globe into the Caribbean. There are some cases now showing up in Florida. West Nile virus, that came from far away, and the list grows. And the more diseases we have that are spread by mosquitoes, and I might say not only mosquitoes, but other vector insects, the more people can. Right here in Tucson, where I live, in southern Arizona, we have a big problem with another kind of insect vector, which is a little creature called a kissing bug. This is a blood-sucking insect as well, but a lot of trouble because it's dangerously allergenic, and it's also the vector of the most important parasitic disease in humans in the Western Hemisphere, which is a disease called Schitt's disease. So we're very interested in that insect here. It's just another case. It's like a walking mosquito. It sucks blood and it transmits disease while it's taking a blood meal.
Do you think these diseases are spreading more because of climate change, or do you think this is something that just goes in waves, or we're just hearing more about it, or is there some other factor?
I think that the scientific community is quite confident that the redistribution, the changing pattern of global distribution of diseases of this kind is due to global climate change. Certainly, just take the examples that I mentioned, not to find mainly to South America, but countries like Argentina and Peru and places in that region. It's now up into Mexico and coming right up to the border of the United States and may well be in the United States already. That's because of global climate change. It's because warming trends, the fact that the winters are less harsh, and deep freezes don't happen in many places the way they used to. Same could be said for mosquitoes and other vector insects, even things that aren't insects like ticks that are vectors. These creatures now have a much wider distribution and therefore ability to spread disease than they did in the days when we had really hard freezes all over the country. Many places now don't see hard freezes anymore. Creatures don't die off in the winter. So yes, I think the biggest factor is global climate factor, is travel. Humans now travel everywhere by air. That didn't happen in the past. And they carry baggage. They carry with them parasites and viral and bacterial and pathogens. And the airplanes can also carry the vector creatures. So there's a very efficient distribution system that we didn't expect. Most of the Chagas disease that we see in the United States is brought in by people who are already infected.
I see. Well, not to end on too ominous a note, let me ask you what you would say to a young person, maybe entering graduate school, who says to you, I love your work. I think about insects all the time, and I want to spend my life studying insects. How would you recommend that they focus? Where do you think the most promising or most intriguing aspects of entomology or study of insects as animal models is likely to go?
Well, I want to encourage any person who's that these are the most species, most prominent fauna on the planet. These terrestrial arthropods, and they do lots of good things for us, pollinating our crops with free pollination services and other good things. At the same time, as we've been discussing they do lots of harmful things, not only for plant crops, livestock, and the love to these creatures and have the courage to do so is that there isn't a single insect that's ever been solved. We have never fully controlled any insect problem. So the opportunities are huge for culture, pollination services of endangered pollinators, because we're killing bees, we're killing other pollinators inadvertently. There's just huge opportunity. The key answer to the question is a general answer, and that is follow your passion. I would tell any graduate student, any undergraduate student, don't do what you should do. Do what you want to do. You love it. That's why I got into insects. I wasn't trained to do this. I discovered it when I was a graduate student working on bacteria. And I decided this is so interesting. That's what I want to do. I don't care whether it's going to be hard to find a job or difficult to get grants. I did it because I love it, and I still do more than 55 years later. So my answer to any student listening is follow your passion.
Well, I think we can't find a better note to end on than that. That's a great endorsement of study in general, and as you say, follow your passion. That's great to keep in mind. Well, Dr. Hildebrand, thank you so much for joining us, and we've really enjoyed talking with you and hearing about just a few aspects of the many things you've studied. And this concludes our Google Hangout for today. And anyone who is interested in upcoming events should check our Google Plus page for the schedule, which will be posted there. Thank you very much.
Thank you.